Influenza, the virus that causes the flu, is one of the most common infectious diseases. Now, there are three types of influenza that infect humans, called Type A, Type B, and Type C, each one with a slightly different genome and proteins. Influenza belongs to the virus family Orthomyxoviridae, and Type A and B have genomes that are made up of 8 RNA segments, whereas Type C has a 7-segment RNA genome, with each segment containing a few genes. Now, Type A, the most common type of influenza virus, can be further subdivided based on 2 of the glycoproteins on its protective envelope surface, H protein or hemagglutinin and N protein or neuraminidase. Hemagglutinin and neuraminidase can vary a bit in their structure, so different versions are identified by a number. For example, Type A, subtype H3N2, sometimes just called H3N2, has hemagglutinin number 3 and neuraminidase number 2 on its surface. H3N2 and H1N1 are the most common Type A subtypes to infect humans, but they both also infect various animals. To give the full name of a virus, we use the type, the original host that it came from, the location where the virus was first identified, which is usually a city, the strain number, the year of origin, and for Type A influenza, the subtype named by the H and N glycoproteins. For example, an H1N1 Type A flu virus of duck origin from the city of Alberta, Canada, that is the 35th strain discovered in 1976, would be called A. duck Alberta 3576 H1N1. Type B influenza is less common, and it only infects humans and doesn't mutate as often as Type A. Type B influenza only has a few types of H and N glycoproteins on its surface. Therefore, the naming pattern is similar to Type A influenza, but without the H and N subtype included at the end, or the host type, since it only infects humans. For example, a Type B virus found in Yamagata, Japan, which is the 16th strain discovered in 1988, will be called B. Yamagata 1688. Finally, there's Type C influenza, which only has one species, and is the least common and least likely to mutate of the three. Influenza C usually causes mild disease in children, and unlike Type B, it can infect both humans and pigs. Rather than hemagglutinin and neuraminidase, Type C influenza uses a hemagglutinin esterase fusion protein to enter and exit cells. So Type C influenza is named without the H and N subtype, similar to how Type B is written. For example, a Type C virus found in Sao Paulo, Brazil, which is the 37th strain discovered in 1982, would be called C. Sao Paulo 3782. Of the three types, Type A is the most common and causes the most severe illness. One reason is that the virus has a tendency to mutate its H and N glycoproteins during replication, and this allows daughter viruses to form that are slightly different from one another and from the parent virus. Over time, if enough of these small changes happen, even if somebody's immune to the original virus, the mutated virus might have H and N glycoproteins that are different enough to allow it to evade antibodies, and therefore infect someone who is immune to previous strains. This process is called genetic drift, and is why individuals can get sick from influenza year after year, or from 2 different strains of influenza in the same year. In addition, a process called antigenic shift is where sometimes a virus will be circulating among animal populations like pigs or chickens, and then it'll suddenly change in a way that allows the virus to infect humans as well. This happens when the same cell, let's say a pig cell, gets infected with 2 similar flu viruses. For example, a flu strain that usually infects humans, and one that usually infects pigs. Since the viral genome is in segments of RNA, the pieces might reassort, or mix, allowing new viruses to have a mix of RNA segments in them. Reassortment results in viruses that have entirely new hemagglutinin, neuraminidase, or both. When a virus is produced that can infect human cells and has entirely new proteins, people have little or no protection against it, and it can rapidly spread through the population. This antigenic shift process is responsible for 3 major influenza pandemics in the 20th century, including the Spanish flu in 1918, which killed 3% of the world's entire population at the time. Now, the flu is transmitted when an infected person sneezes or coughs, which spreads thousands of droplets containing the virus into the local area, up to about 2 meters or 6 feet away. These droplets can then land in the mouths or noses of people nearby, or get inhaled into the lungs. The virus can also survive on surfaces for a few hours, so it's possible to get the virus by touching a surface, like a contaminated doorknob, and then touching your own eyes, nose, or mouth. When the flu virus enters the body, it uses hemagglutinin to bind to sialic acid sugars on the surface of epithelial cells in the upper respiratory tract. Once bound, the cell swallows up the virus in a process called endocytosis. That viral RNA is negative sense, which means that each piece first has to be transcribed by RNA polymerase into positive sense mRNA strands, before it can be translated into proteins and assembled into new viruses. These viruses then leave the cell by simply budding out from it by using the neuraminidase, which cleaves the sialic acid sugars in the membrane, releasing the newly created viruses from the cell. Influenza symptoms start 1 to 4 days after infection and include fever, headache, runny nose, sore throat, and a cough. Most of these symptoms get better within a week, but the cough often persists for up to 2 weeks. There can sometimes be complications, though, like acute otitis media, bronchiolitis, croup, sinusitis, and pneumonia, including antibiotic-resistant strains caused by Staphylococcus aureus and Streptococcus pneumoniae. The highest risk of complications is among groups like young children under 6 months of age, pregnant women, adults over 65 years old, and those with chronic medical conditions like chronic heart or lung disease. Young children in particular are more likely to have neurologic complications like encephalitis and febrile seizures. One unique complication is Reye syndrome, which is a condition where after taking aspirin while fighting off a viral infection like the flu, a child develops encephalopathy and liver disease. Although less common, all of these complications can also happen in previously healthy individuals as well. Although most people don't show symptoms until a few days after infection, the disease can be spread to other people 1 day before symptoms even start, and up to a week or 2 afterwards. This means that a person can pass the flu to someone else even before they have symptoms and feel sick. Now, there are a lot of tests used to detect if an individual is infected with influenza. Rapid influenza diagnostic tests can detect the flu virus from nasal secretions within minutes, but often vary in their reliability and often detect a certain type of influenza, but not the specific strain of influenza A, like H1N1, for example. There are 2 more accurate and reliable ways to identify influenza. The first is a viral culture, which involves growing the virus and identifying it. And the second is to use polymerase chain reaction to detect even a tiny amount of viral RNA. Medical treatment for influenza is usually used for high-risk patients, or if the illness is severe. There are 2 classes of medicines. The first is neuraminidase inhibitors, which inhibit neuraminidase, and stop the virus from breaking out of the host cells. The second are M2 proton channel inhibitors, which are made of a compound called adamantane, and prevent viruses from replicating inside the host cell. It turns out, though, that the virus's M2 gene mutates frequently, which allows it to become resistant to the M2 proton channel inhibitors. Now, the most effective way to prevent influenza is through vaccination, and there are 2 that are commonly offered. One option is a trivalent inactivated influenza vaccine, or TIV, which is a killed virus that's injected into a muscle. And the other is a live attenuated influenza vaccine, or LAIV, which is a weakened virus that's sprayed into the nose, so it can infect the epithelial cells. Both of these are actually trivalent, meaning they have a mixture of the 3 strains that are predicted to be the ones that'll dominate the coming season. This prediction is based on what strains typically dominate year after year, like H1N1, H3N2, and influenza B, as well as real-time data on which strains are circulating during the winter season on the other side of the planet, since these ones are likely to spread and cause trouble. This is because countries in the northern hemisphere, like the US and Canada, experience winter and therefore the flu season at the opposite time than other countries, like Australia, in the southern hemisphere. Now, there are a few risks associated with getting the flu vaccine. Most flu vaccines are produced by growing the virus inside of a chicken embryo, and therefore the vaccine contains small amounts of egg protein, which can be a problem for individuals with a food allergy. Guillain-Barré syndrome, an autoimmune disorder of the peripheral nervous system, is another side effect believed to be associated with the influenza vaccine. Lastly, children under 6 months old are in the pediatric group at the highest risk of serious influenza complications, but they're too young to get the flu vaccine. That said, the best way to protect these young infants is to make sure their caregivers are vaccinated, which creates a natural barrier to infection. On average, the flu vaccine reduces the risk of illness by half, roughly falling from 10% to 5% in terms of likelihood of getting sick over the entire winter season. And two factors play an important role in determining if the flu vaccine will protect a person from illness. First, a person's characteristics, like being in a high-risk category, can make that person more susceptible to the virus despite being vaccinated. Second, the vaccine has to match the circulating flu virus of the season. Since flu viruses are constantly mutating, the flu vaccine formulation is reviewed each year to keep up with any changes. Finally, it's important to be vaccinated each year before the start of the flu season to ensure protection against the circulating virus. Now, getting vaccinated is super important for both yourself as well as the community, since if the majority of a community is vaccinated against influenza or any other contagious diseases, it actually protects the other members of the community, even those few that aren't vaccinated. Alright, so let's say that nobody's vaccinated, and one person gets the flu and then comes in contact with two other people. Those two people then get the flu, and each spread it to two other people. And this chain reaction goes on, maybe even causing an influenza outbreak in that community. But now let's say that the majority of people are vaccinated. In this case, the chain reaction can't spread through the whole community. So even if a few people aren't vaccinated, they're still protected. This is called herd immunity, because the herd's protecting the weakest members. This is especially important for people who can't get certain vaccines, like infants, pregnant women, or immunocompromised individuals, because they still get some protection when the spread of contagious disease is contained. Alright, as a quick recap, influenza, also known as the flu, is a common infectious disease and can spread from person to person through tiny droplets in the air or on surfaces. Most people recover from the flu without complications, though sometimes antiviral treatments can be used. And the most effective way to prevent the flu is through seasonal flu vaccination.